Proteins. Proteins are a group of macromolecules. Elemental composition is C, H, O, N and sometimes S. They're made up of many small monomer units called amino acids, joined together by condensation reactions. The bond formed is known as a peptide bond. There are 20 different types of amino acids in nature. Amino acids. At the center of the amino acid is an asymmetric carbon. Each amino acid is made up of an amino group, carboxylic acid group, hydrogen atom, and AR group. Except glycine, others have an asymmetric carbon. The R group is called the side chain, while other groups are in the backbone. The R group will be responsible for interactions of proteins depending on whether it is polar or not. And also the R group might contain sulfur or selenium. Formation of proteins. Amino acids are joined by a reaction between the amino group of one amino acid and the carboxyl group of another. They join in a condensation reaction, and a molecule of water is released. A peptide bond is formed when two amino acids join, and a dipeptide is the result. More and more amino acids join to form polypeptide chains. The carboxylic group of amino acids have acidic nature, while the amino group has an alkaline nature. Bonds in proteins. There are 4 other bonds than peptide bonds. They are hydrogen bonds, disulfide bonds, ionic bonds and other weak interactions. Hydrophobic or van der Waals bonds. Hydrogen bond. Bonds formed between negatively charged oxygen in carboxylic groups and positively charged hydrogen atoms of amino groups. Hydrogen bonds break easily and reform if pH or temperature conditions change. Disulfide bond. Disulfide bonds form when two cysteine molecules are close together in the structure of a polypeptide. An oxidation reaction occurs between the two sulfur-containing groups, resulting in a strong covalent bond known as a disulfide bond. These disulfide bonds are much stronger than hydrogen bonds. Ionic bonds. Ionic bonds can form between some of the strongly positive and negative amino acid side chains. They are strong bonds. Structure of proteins. There are 4 structures of proteins. They are primary, secondary, tertiary and quaternary. Primary structure. The primary structure of a protein is the sequence of amino acids that make up the polypeptide chain, held together by peptide bonds. Secondary structure. The secondary structure of a protein is the arrangement of the polypeptide chain into a regular, repeating three-dimensional, 3D, structure, held together by hydrogen bonds. Most fibrous proteins have this type of structure. Tertiary structure. The amino acid chain, including any helices and pleated sheets, is folded further into complicated shapes. Hydrogen bonds, disulfide bonds and ionic bonds between the R groups of nearby amino acids hold these 3D shapes in place. Globular proteins are an example of tertiary structures. Quaternary structure. Found in proteins consisting of 2 or more polypeptide chains. Examples include some very important enzymes and the blood pigment hemoglobin. Denaturing of proteins. Loss of the 3D shape of the protein due to changes in conditions such as temperature or pH. Fibrous proteins. They have little or no tertiary structure. They are long, parallel polypeptide chains with occasional cross-linkages that form them into fibers. Found in connective tissue and tendon and the matrix of bones, as the silk of spiders' webs and silkworm cocoons, and as the keratin. Collagen. Collagen is a fibrous protein that gives strength to tendons, ligaments, bones and skin. Its quaternary structure has 3 polypeptide chains. The 3 polypeptides are chains, often found together in fibrils that are held together to form collagen fibers. In the genetic disease osteogenesis imperfecta, the collagen triple helix does not develop properly. Globular proteins. Globular proteins have complex tertiary and sometimes quaternary structures. They fold into spherical, globular shapes. Hemoglobin is one of the best-known globular proteins. Functions of globular proteins. Globular proteins are important in immunity, antibodies. Some act as enzymes and hormones, help to maintain the structure of the cytoplasm. Hemoglobin takes up and gives up oxygen in the tissues. Conjugated proteins. Conjugated proteins are protein molecules that are joined with a conjugated to another molecule called a prosthetic group. Hemoglobin is a large protein with iron as the prosthetic group. It is a conjugated protein, as well as a globular protein. Lipoproteins are formed when proteins are conjugated with lipids. Glycoproteins are proteins with a carbohydrate prosthetic group. Mucus and the synovial fluid in the joints are glycoproteins. Lipoproteins are very important in the transport of cholesterol in the blood. There are 2 types of lipoproteins: low-density lipoproteins and high-density lipoproteins, HDLs. Carbohydrate part of the conjugated protein helps them to hold a lot of water, and also makes it harder for protein-digesting enzymes, proteases, to break them down. Practical skills. Add biuret reagent, ready-mixed 5%, with V, sodium hydroxide solution, and 1%, with V, copper sulfate solution. A purple color indicates the presence of protein. Questions. 1. How are the peptide bonds formed? 2 amino acids undergo condensation reaction and form a water molecule with OH group of carboxylic acid group and H of an amino group. 2. What are the functions of proteins? Act as enzymes and hormones. Help in muscle contraction. Make antibodies. Transport oxygen in the form of hemoglobin. Proteins make structures such as hair, skin, and nails. Give strength to tendons, ligaments, bones, and skin by collagen fibers. Help maintain the structure of the cytoplasm. Mucus is slippery and viscous and helps to reduce friction. 3. Explain the water solubility of proteins. They do not dissolve. Instead, the molecules are so big that they form a colloid. In a colloid, microscopic particles of one substance, in this case protein, are suspended throughout another substance, in this case water. They do not settle, and they cannot easily be separated.